Good afternoon, and welcome to the Florence Mahoney Lecture in the Director's Wednesday Afternoon Lecture Series. I'll just very briefly introduce the history of Florence Mahoney, then have the privilege of introducing Francis Collins, who will in turn introduce our speaker today. Florence Mahoney was an extraordinary woman who was remarkably influential in political circles and directed that sphere of influence in support of NIH over years. Florence died in 2002 at the age of 103. Up until the very last, she was intensively interested in this lecture series in her name. She was adamant that we should focus on only hard science and none of this frivolous stuff, and I think you should be absolutely delighted with our choice of speaker today. I only want to read one quote about Florence Mahoney and published in a biography. Jack Valenti, who is a friend of hers and was an advisor to the Johnson administration, said, when I would tell him, the president, that Florence Mahoney and Mary Lasker, who worked together, wanted to see him, the president would groan. He'd say, oh my God, these two women are going to bankrupt this country, and they usually got what they wanted out of him. And we applaud Florence Mahoney. We look for future champions in this direction. And in that very same mode, it's my privilege to introduce the director of NIH, Francis Collins.
Well, thank you, Richard. Good afternoon to all of you. It is my privilege to introduce the speaker for this year's Florence Mahoney Lecture, and it's wonderful to see such a full auditorium. I think that indicates that maybe people at NIH are interested in stress. I'm sure I don't know why. We are fortunate today to have a speaker, a scientist of considerable renown, who is as close to the holistic model as one could hope, I think, to encounter in terms of the way in which his own research includes detailed analyses at the cellular level of the effects of stress on individual neurons, all the way to understanding how stress affects individuals and social groupings in the work that he has done in East Africa studying baboons. And I'm hoping he's going to tie that all together into a package that will elevate all of our understanding of this important aspect of what it means to be alive and how stress plays out in various ways in terms of health and disease. Dr. Sapolsky has a distinguished academic record. He's currently the John A. and Cynthia Fry Gunn Professor of Biology, Neurosurgery, and Neurology at Stanford. He obtained his PhD in neuroendocrinology from the Rockefeller University. He was previously a MacArthur Fellow. He has received many awards that I won't even try to name, but it's quite an interesting group. Looking through his CV, I encountered hundreds of publications in distinguished places. I can't help but say I also found it kind of enjoyable to get to the end of his CV and see a special category called op-ed pieces, book reviews, prefaces, and articles in embarrassing places. And I'm not sure which ones were supposed to be the embarrassing places. There's a lot of USA Today and some San Francisco Chronicle. I suspect maybe it was item 16 from 2002 in Men's Health, entitled Guerrilla Dating Tactics. You can ask him about that later. So please join me in welcoming Dr. Robert Sapolsky.
Well, you see the drawbacks of being honest on your CV, so take that as a cautionary note. Well, I'm delighted to be here, and thank you for the invitation, and it's always very good to speak at an aging context in honor of someone who lived to 103, so I'm hopefully going to pick up some sort of pheromones from Florence Mahoney, or maybe not, maybe that's the wrong way to state it. Anyway, but starting off, I know many people here, and based on that, I'm willing to sort of extrapolate a bit and bet that nobody in this room has leprosy. I would guess nobody here is going to be running to the bathroom every 10 minutes because of their dysentery. Probably nobody here is teeming with liver parasites the size of your fists. Probably nobody here is a normal mammal. You don't worry about smallpox, scarlet fever. Few of us come down with malaria during the rainy season. Few of our mothers died in childbirth. Nobody in this room is malnourished. We're not like normal mammals. We don't die like normal mammals. Run-of-the-mill, off-the-rack mammal, what happens? You drink some contaminated water, and you're dead 2 days later from dehydration. What do? We spend 75 years having our bodies go to hell on us. And this is wonderful news. This is wonderful news because we succumb to westernized disease, diseases of slow accumulation of damage. And when you look at the ones that do us in, these are very different diseases than the ones that got our ancestors. A world now where we live well enough and long enough to fall apart with cardiovascular disease and diabetes and cancer and neurodegenerative disorders. And this is an extremely different world of who is healthy and who's sick. 20,000 years ago, you're some hunter-gatherer, you have screwed up big time, you have just eaten some reed buck riddled with anthrax, and the medical outcome's absolutely clear. You've got like a 3-day life expectancy. These days, you've screwed up because you've decided a diet of red meat and saturated fats and maybe a drink every other day or so constitutes a healthy diet, and it's not at all clear what the outcome's going to be. You may be dead in your grave at 50, or you may be running marathons with your grandkids when you're 85. And in lots of ways, the central question in westernized medicine these days is, why do some of us last to 50 and some to 85? Now, some of it's got to do with nuts and bolts biology and what your liver does with cholesterol and, that whole world. But some of it's got to do with questions that nobody ever had to think about before in medicine. Utterly bizarre questions like, what's your psychological makeup? Or what's your social status? Or how do people with your social status get treated in your society? Or even something as bizarre as, when you're feeling unloved, do you eat more carbohydrates? Figure that one out and you've solved half the cases of diabetes in this country. This is a very different world that we've entered. And when we look at these diseases that do us in, these diseases of slow accumulation of damage, these are predominantly diseases that can either be caused by or be made worse by stress. Most of us in this room will have the profound westernized luxury of dropping dead someday of a stress-related disease. Nonetheless, it's a good thing that this happened later rather than sooner. What I'll be focusing on today is what is by now a 60, 70-year-old literature showing how stress could adversely affect your body, pathogenic processes all over the place. We're perfectly comfortable by now with the notion that stress can do bad things to your blood pressure and your stomach walls and your gonads, all of that. What I'll be focusing on is what is now a 20, 30-year-old story showing that in addition, stress can have some very adverse effects in the nervous system, even down to the level of neurotoxicity. What I'll do is focus on what we understand now to be the cell molecular biology of how things go wrong, and just when one is about to think perhaps this is only bad news for cultured neurons, showing the increasing evidence that this is in fact pertinent to our own human brains. Okay, at that point, if you're still awake, you should be depressed as hell because it's nothing but bad news between now and then. What I will then focus on is a very reductive realm of potential good news, the capacity to do a rational intervention in the life of a neuron, the possibilities of doing gene therapy in order to save neurons from these adverse effects of stress. Finally, in the last third, what I'll focus on is a much more integrative level, getting at the sheer issue of why do some of us succumb to stress more readily than others, and using, as you'll see, a rather unlikely study subject. Okay, so starting off, stress. When saying stress, not meaning some sort of undefined state, but rather a class of stress hormones that will be the villains throughout, glucocorticoids, adrenal steroids released in response to stress, human version, hydrocortisone, also known as cortisol, rodent version, of great significance to the bad news to come. Many millions of prescriptions written each year for synthetic glucocorticoids, such as prednisone, dexamethasone. And what we have, classic neuroendocrine cascades. Something stressful occurs, the brain mumbles to the pituitary, mumbles to the adrenals, and out come glucocorticoids. Glucocorticoids are the damaging hormones in the story. And 25, 30 years now into this finding, you sit down your basic endocrinologist and they think this is the most ridiculous thing they've ever heard. Because if you don't have glucocorticoids, you are dead. They are essential for life. Where do they get off? Damaging your brain cells. And to begin to appreciate that, what you need to do is get a sense of what glucocorticoids are doing, because they absolutely encapsulate the double-edged sword of the stress response. What you do with glucocorticoids and the other players in the stress response, what you do with it is exactly what your body needs done if you're getting stressed like a normal mammal. You were a zebra, a lion has leapt out, ripped your stomach open, your innards are dragging in the dust, and you still need to get out of there. And glucocorticoids are essential to this process. They mobilize energy, delivering energy to your exercising muscles, diverting it from fat, from glucose, breaking down glycogen, breaking down triglycerides. You increase your cardiovascular tone, increase that glucose and oxygen levels in your bloodstream as fast as possible, get it to your thigh muscles in 2 seconds instead of 3. You're that much more likely to survive. During stress as well, you turn off all sorts of long-term building projects in the body. And this makes perfect sense. If there's a tornado due this afternoon, you don't spend the day outside gardening. You don't worry about the long-term projects until you know there's a long-term. You shut down digestion. If you are the lion chasing after a zebra, you don't have a full stomach. If you were a zebra running for your life, the energy you're mobilizing, this first line, is happening in like 2 seconds from your fat cells. Digestion is slow, it's expensive, it takes forever. You're trying to avoid being somebody's lunch. Don't worry about digesting breakfast. And we all know the first step of this. Suppose you get a little bit stressed speaking in public. What happens? Your mouth gets dry. You've stopped secreting saliva, the first step of shutting down the system. In addition, you shut down growth, you shut down reproduction, big, expensive, optimistic things to be doing with your body, and this is no time for it. You're running for your life, there's a lion 2 steps behind you. You know, ovulate some other time. Don't do it right now. Hit puberty next week, grow antlers tonight, don't even think about sperm. Hallmark of the stress response, you shut down growth, you shut down tissue repair. Every sex hormone on earth disappears from the bloodstream. Do it later if there is a later. Continuing all the way down to stress hormones, glucocorticoids getting into the brain, and short term, they do wondrous things. They sharpen cognition, they lower your sensory thresholds, flashbulb memory, the where were you. Every one of us in this room will remember forever exactly where we were when we heard the news that Britney Spears had shaved her head. So you need some sort of mechanism saying, this is important, file it away. Look at all the stuff you're doing here on the left. You're mobilizing energy, you're delivering where it's needed, you're shutting off the unessentials, you're thinking more clearly, absolutely essential if you are going to be a normal mammal being normally, acutely, physically stressed, and glucocorticoids are central to this. What you see on the right, though, is what if you secrete them too often, too long, for purely psychogenic reasons, psychosocial ones, and we see a whole array of the pathologies of an excess of stress and excess of glucocorticoids, not coincidentally, some of the leading causes of disease of Westernized lifestyle. So we have this double-edged sword. If you plan to be stressed like a normal mammal, you had better secrete glucocorticoids. If you are going to be stressed like a cognitively sophisticated primate like us, which is to say chronic psychosocial stress, glucocorticoids are bad news. Now, it's in that context that we can appreciate this last line, this ability of chronic glucocorticoid exposure to begin to damage the nervous system. And again, this is a literature going back decades. You disrupt the sort of memory specialized by a part of your brain, the hippocampus, explicit declarative memory, chronic stress, chronic glucocorticoids, it's impaired. And all the building blocks of it are as well. Non-neurobiologist, LTP is long-term potentiation. It's probably what's going on between your synapses when learning occurs. Essential in the hippocampus, glucocorticoids and stress disrupt it. Over the course of days to weeks in a rodent, stress glucocorticoids causing retraction, dendritic atrophy, disconnecting circuits. Probably the biggest revolution in neurobiology for the last 2,000 years. If you took an intro neuro class, they taught you the adult brain doesn't make new neurons. That is not the case. It is centered in the hippocampus in response to learning, exercise, stimulation, and the surest way to shut it down, stress and glucocorticoids. Finally, as I'll be focusing on, the ability at an extreme of glucocorticoids to overtly kill hippocampal neurons or to endanger them, to make them less capable of surviving a neurological insult. So the question becomes, where do these hormones get off damaging your hippocampus? Why the hippocampus? And this was appreciated in the late 60s, work of Bruce McEwen and colleagues, Rockefeller University, who mapped the first steroid hormone receptors in the brain. And as I understand it, virtually by random, he started with glucocorticoids, and back came autoradiograms like these, screaming the hippocampus, the target site for glucocorticoid action. And from that day on, if you're going to think stress and the brain and glucocorticoids, you got to start with the hippocampus. Over the subsequent years emerged a picture of glucocorticoids being able to damage the hippocampus. And every one of these slides is a dozen years' work from various labs, but a literature coming out then showing lots of stress, lots of glucocorticoids, and you make a hippocampus get older faster. More dead neurons, more reactive gliosis, more of the associated memory problems. Conversely, go in there and any of a number of different techniques decrease the cumulative glucocorticoid exposure, and you slow down the hippocampal aging. And you put the pieces together of cumulative glucocorticoid exposure over a rat's lifetime, a very dramatic pacemaker of the rate of hippocampal aging. Okay, so how does this work? How are these glucocorticoids killing hippocampal neurons? And it struck me in the mid-80s or so that in fact glucocorticoids didn't need to be killing hippocampal neurons. All they needed to be doing was messing them up in some way. All they needed to be doing was taking a hippocampal neuron and leave it dangling on the edge of some cliff. At that point, if nothing else happens, everyone lives happily ever after. On the other hand, if at that point you push the neuron with something else, it's that much more likely to go over the edge. And what this notion of endangerment predicted was all sorts of neurological insults that damage the hippocampus should be made more damaging when there's a lot of glucocorticoids on board. And this turned out to be the case with a vengeance. All sorts of the leading necrotic insults to the hippocampus, the more glucocorticoids in the circulation at the time, the more of an acute stress history, the more dead neurons afterward. Ischemia, stroke, hypoglycemia, sustained seizures, oxygen radical generators of great disturbing relevance, the beta amyloid peptide, the rogue peptide in Alzheimer's disease, GP120, the glycoprotein from the AIDS virus, responsible for some aspects of AIDS-related dementia. What we got here are an enormously varied number of ways of doing in a neuron, all of them made worse by glucocorticoids. Whatever this cliff of vulnerability is about, it's a very generalized one. So what is it about? What's going on here? What you then begin to see is all sorts of possibilities. How are glucocorticoids doing this? They could be changing cerebral perfusion rates and insulin levels and 11 different things in the periphery. The surest way to show that it's not a phenomenon of the peripheral physiology is get rid of the peripheral physiology. Do all the same thing in vitro models of these insults, and you see the same thing, insult, and the more glucocorticoids on board, the fewer surviving neurons. And using these in vitro approaches, you could do your basic endocrine homework and ask, for example, do you see endangerment by glucocorticoids like this in other brain areas, cultures from cortex, hypothalamus, cerebellum? You do see it in other areas, nowhere near as dramatically as in hippocampal neurons. This is the most sensitive area. Second question, what about other steroids? What about specificity? Do you see endangerment by progestins, androgens? No, not at all. How about estrogen? Of great relevance to about 30 minutes from now. Not only does estrogen not make things worse, estrogen is protective. We'll come back to that. Finally, for those who care about such minutiae, there's two different types of receptors for glucocorticoids. It's the GR that mediates this. This is classical, boring endocrinology. This is a hormone working in a specific way through its receptor so that whatever you now throw at the target cell, it's going to be in a more fragile state. So what is this about? How are glucocorticoids endangering? And it struck me by the late 80s or so that there was one thing in common. That slide I just showed you, all those different neurological insults, so heterogeneous, they all ultimately have one thing in common, which is they ultimately constitute an energy crisis. Either the neuron can't generate enough energy, hypoglycemia, hypoxia-ischemia, or the neuron has such insanely high energy demands, sustained seizure, that there's an energy crisis. So looking at that struck me that maybe what this generalized cliff of vulnerability is about is in some way an energetic one. Now, what this predicted was a very simple experiment, which is you should be able to take one of these insults that causes like this much damage. Now, insult plus glucocorticoids, you get this much damage. Insult glucocorticoids and feed the neurons something extra, supplement them with excess energy, and you will subtract out the glucocorticoid endangerment. And that's precisely what you wind up seeing, all sorts of ways of demonstrating this. Here, just one version, a particular insult. Insult alone, this much hippocampal damage. Insult plus glucocorticoids, far more damage. Insult, glucocorticoids, and feed the neurons mannose. Mannose, this monosaccharide, it has its own transport system in the brain, works perfectly well. Away goes the endangerment. Try the same thing with fructose. No luck, doesn't get past the blood-brain barrier. Use one of the only other things neurons can eat, ketones, subtracts as well. It's an energetic endangerment. So by the early 90s, we had figured out the mechanism for this. And as neurobiologists, we were thrilled because we were among the first to see this. And as endocrinologists, we were totally bored because everybody had known this forever. It's got something to do with the logic of what you want to do with your circulating glucose when you're stressed. You're running for your life from the lion. You've just done gluconeogenesis in your liver. You've dumped glucose in your bloodstream for your exercising muscle. What would be the stupidest thing on earth now to do with your circulating glucose? Store it away in your fat cells for next spring. Store it away in your liver. Store it anywhere. And for 50, 60 years, it's been known glucocorticoids inhibit glucose uptake and storage throughout the body, except for exercising muscle, a strategy for diverting it to where it's needed. So we went and looked at, in the hippocampus, the exact same thing. Glucocorticoids inhibiting glucose uptake by way of pulling glucose transporters off of the cell surface, less glucose uptake at that point. Is this a big effect? Not at all. Take a fat cell and glucocorticoids and you knock out like 80% of glucose transport. Do it with a neuron, you knock down maybe 20%. That's not enough to kill a neuron, but you're not trying to explain killing a neuron. You're trying to explain making it a little bit queasy and fragile. And what you see then is along comes an insult, and with glucocorticoids, ATP levels drop sooner, mitochondrial potential collapses faster. You have a neuron that is more energetically vulnerable at this point. So what goes wrong at this point? And I put up here this magnificent 3,000-year-old Egyptian hieroglyphic showing the synapse, which I do need to replace one of these days. But what we see here is the basic workings of excitotoxic, necrotic, neurological neuron death, the centerpiece of all of it being the release of a class of neurotransmitters, excitatory amino acid neurotransmitters, most notoriously glutamate. Glutamate. If you want to learn and do that LTP stuff, you better release glutamate in your hippocampus, but all these insults involve a vast excess of glutamate causing tidal waves of calcium mobilization, causing downstream generation of oxygen radicals and cytoskeletal breakdown and all sorts of bad stuff. All of those neurological insults involve this pathway, tremendous amount of work at blocking various receptors, chelating calcium. This is the central dogma of acute necrotic neuron death. Critically, every step in this pathway is made worse if you're running out of energy. At the presynaptic end, the neurons are more readily depolarizing, they dump more glutamate. Even more importantly, they don't have the energy to run these high affinity reuptake pumps. More glutamate sticks around longer. More calcium rushes in and not enough energy to pump them out. And what you see is all of these steps are energy dependent. Now at this point, you don't need to be a brain scientist to come up with a scenario that, insofar as glucocorticoids make all of these energy dependent insults worse in an energy dependent manner, maybe what they're doing is by inhibiting glucose uptake, the neurons are simply less capable of doing every step along the way. And in this badly doctored hieroglyphic, here was the 90s in my lab showing this is precisely what occurs. Every single step is made worse with glucocorticoids on board. More glutamate sticks around longer, more calcium sticks around longer, more of all the downstream steps, all of them reversible with excess energy. So in lots of ways, that's kind of where this story is at this point. Glucocorticoids hammering away at these neurons, making them more fragile. Neurons, however, do not just sit there and get pummeled by these insults. There's a variety of defenses they can mobilize. They upregulate heat shock proteins, antioxidants. There are retaliatory neurotransmitters that inhibit glutamate release. There's a whole bunch of these defenses. Collectively, they're somewhat protective. Collectively, they're really expensive. And thus, you know the next sentence I'm going to say. Not only do glucocorticoids make the insults more insulting, they impair the capacity of neurons to mobilize these defenses. So that's where the story is at, except for one complication, and this is one that is always floated around. If you're oriented towards the world of neuron death, a dominating theme these days is inflammation. Unhappy brains have chronic inflammatory states. Inflammation plays a big role in the neuron death after both acute insults and slow neurodegenerative diseases. In a broad, sustained way, big-time inflammation, bad news in the brain. Glucocorticoids and inflammation, glucocorticoids are anti-inflammatory. Everyone learns that in kindergarten. Glucocorticoids are anti-inflammatory. Thus, glucocorticoids should be decreasing damage, not making things worse. And for about 15 years, sort of my next to last paragraph, my RO1s would always hand wave like crazy on this one, saying, well, insofar as you have glucocorticoids making things worse for the neurons, but decreasing the inflammation, ooh, you got the arrows pointing in two different directions. Which arrow is stronger? Put the pieces together in a whole system, and if you've got more dead neurons coming out the other end, it means the endangering component is more powerful. Then about a half dozen years ago, I had a first person in my lab, an immunologist, decide to actually look at this, to finally explicitly show that, yes, this arrow isn't as powerful as this one, and something very interesting came out instead, something that has now been seen in a number of labs, which is there's all sorts of domains in the acutely injured nervous system where not only are glucocorticoids no longer anti-inflammatory, they can worsen inflammation. They are pro-inflammatory. And this has now been shown, my group, on a number of different levels. Glucocorticoid stress increasing the amount of inflammatory cell migration to the injury site, more production and release of inflammatory, pro-inflammatory cytokines. A absolutely textbook thing that glucocorticoids do is inhibit the activity of NF-kappa B, pro-inflammatory transcription factor. What they do in the injured hippocampus and cortex is they increase the activity of NF-kappa B. This whole pathway here exacerbated, and we're beginning to believe what it involves is oxidative damage to a cofactor of the glucocorticoid receptor, which changes receptor interactions with NF-kappa B. And for us, most remarkably, is 1 inch away in the rest of the brain, classically anti-inflammatory. So this is a new disturbing piece of the story. Okay, so all sorts of this is bad news. Bad news for rats, for mice, for cultured hippocampal neurons. What about us? What about the primate brain? Now, the primate brain has the hippocampus, even though it's spelled differently in the primate, but up until a number of years ago, about a decade ago, the entire literature of asking, well, is this pertinent to the primate hippocampus, the human one, consisted of a number of studies that came out of my lab looking at captive primates and showing sustained social subordination stress, and you get the same thing as in the rodents. You get hippocampal damage at the EM level, same exact picture. One would conclude this may be a bad sign for stress. Then follow-up studies surgically implant cortisol-secreting pellets in the hippocampus of monkeys, come back a few years later, same bad thing. This suggesting that maybe all this bad news is in the ballpark of being pertinent to humans. In the last 10 years or so, what's become clear is it is far closer than merely in the ballpark. A number of different bodies of evidence, 3 of the strongest now. Cushing's syndrome. Cushing's, most will know, any of a number of disorders resulting in wildly elevated glucocorticoid levels. You get every pathology on the right side of that chart back when, including memory problems, hippocampal-dependent memory problems, often known as Cushingoid dementia. And about a decade ago, Monica Starkman and colleagues, University of Michigan, showing atrophy of the hippocampus, only the hippocampus and Cushing's. The more elevated the glucocorticoid levels, the more atrophy, the more memory loss. Bad news. Is this due to dead neurons? Almost certainly not, because what they then showed was go in and surgically get rid of the tumor, and over the next year or so, hippocampus comes back to normal volume and cognition normalizes. Next one. PTSD, post-traumatic stress disorder, combat vets, childhood abuse, and by now an extensive, well-replicated literature showing atrophy of the hippocampus, preferentially so the more severe the trauma history, the more atrophy, the more memory problems. Is this one reversible? As far as I know from the literature, no, but that may well be because PTSD actually is never cured. People learn to manage it. A huge incendiary question in that field right now is whether the trauma causes atrophy of the hippocampus or whether a small hippocampus sets you up for being more vulnerable to trauma. At least some of the evidence shows that trauma can lead to hippocampal atrophy. Finally, of alarming pertinence to like 15% of us in this room, those of us destined to have a major league depression at some point or other, depression in about half of patients associated with highly elevated glucocorticoid levels, and the literature now showing atrophy of the hippocampus, the more severe the depression history, the more atrophy, the more memory problems, and at present, the bulk of those studies show that when the depressions have been normalized, you do not get normalization of the hippocampal volume. Okay, so as promised, you should be utterly depressed by now. Okay, let me make it even worse. More bad news. So what does this tell us also about the hundreds of thousands of individuals getting high-dose corticosteroids each year for their rheumatoid arthritis, their lupus, all of that? Do they get memory problems? Well-documented steroid dementia. And the first good case control studies are now showing that for the same inflammatory disease, the more of a history of treating it with steroidal anti-inflammatories rather than non-steroidal, the more cognitive problems. Finally, what this also is telling you is if you go and have yourself a stroke, please mention to your clinician not to give you corticosteroids at that point. Typically done at that stage in some of the less than leading medical institutions in this country to take down the post-stroke edema, and it's been clear for 30 years that it doesn't do a very good job of taking down the post-stroke edema, and it tends to worsen the neurological outcome. So the best people in the field for decades have been saying, don't use steroidal anti-inflammatories for post-stroke edema, use non-steroidals. More evidence supporting this. But the worst news in some way is go and have one of these neurological disasters and you don't need your neurologist to give you glucocorticoids at that point to get into trouble. You're secreting boatloads of the stuff on your own. Somebody who has just had a concussive head trauma, grand mal seizure, cardiac arrest, some of the highest levels of glucocorticoids you will ever see in a mammal, and they make the damage worse. How do you know? Take a rat and adrenalectomize it right after one of these insults, and there's less damage afterward. In other words, what we've been taught to think of as the typical damage you get after these insults in the hippocampus is damage made worse by the fact that our bodies stupidly go and have stress responses at the time. I mean, look at how bizarre this is. You get chased by a predator, you secrete glucocorticoids, divert energy to your thigh muscles. Saves your life. Psychological stress, you're on a blind date. You secrete glucocorticoids and divert energy to your thigh muscles. Probably irrelevant. You have a stroke. You have a stroke and you divert energy to your thigh muscles. Totally maladaptive. This is very, very useless from a neurological standpoint. So what can you do about this? Obviously, you can't adrenalectomize somebody just because they've had a seizure, but all sorts of things can be done. There's a whole class of drugs, well documented, well studied in various endocrine clinics for years, used in other domains, that are steroidogenesis inhibitors, and they will shut down glucocorticoid secretion for 12, 24, 48 hours or so. And my lab and others showing get it into the animal right after the insult, stop the glucocorticoid secretion, you decrease the damage. But what if you're too late for that and you've secreted the hormones already? Next obvious option is get a glucocorticoid receptor antagonist, block the receptors, and the very renowned progesterone receptor blocker, RU486, is also quite good at blocking glucocorticoid receptors, a literature now showing that can be protective as well. But what if you're too late for that? They've already bound to their receptors, they're already messing up energetics, and what you see here is the strategy showed earlier, energy supplementation. But what if you're too late for that? And you're already now in the realm of more glutamate and more calcium, more oxygen radicals, and you have now entered the central arena of neuron death. And what I put here is not facetious, but the transition instead to the next section, go and build yourself a better neuron. And from the early 90s, the main focus of my lab has been doing so with gene therapy strategies. Okay, at this point, it's not hard to come up with a shopping list of what sort of genes you might want to overexpress in a neuron going through one of these acute insults. What, of course, has always been the challenge is how are you going to do it? For the very simple reason that the gene therapy techniques that like you could do in your liver that are so easy, like high school kids doing for science fair projects, you can't use any of that stuff in the nervous system because the brain's heterogeneous and the brain's inaccessible. And most of all, because the brain's post-mitotic and neurons, most of the viral approaches, retroviral ones, are taking advantage of cell division. What you're left with is a relatively small family of neurotropic viruses that are able to infect non-dividing cells. And since over these years, my lab is focused on using a number of these to deliver the genes in question. Okay, so starting at that time, early 90s, what genes would we want to overexpress? And starting off right then, the first gene we chose, not very surprising, was one that focused on everything that goes wrong in all of these insults. Again, no matter what the acute insult is, a little bit of extra energy is going to help. So the first one starting with overexpressing the glucose transporter. So how do you show this is useful? First, you do your basic molecular homework. Can you find a vector that's safe? Does it express? Is the protein made? Does the protein wind up in the right place? You do all that, good. Next step, if you're overexpressing a glucose transporter, you better be able to show that these are neurons that now take up more glucose. And you can show this in vitro, in vivo, sort of controls in vitro, either mock infection or infection with a vector only with a reporter gene, showing thus in vivo 2-deoxyglucose mapping, great. Neurons are taking up more energy. If you've gotten that far, you now better be able to show that these are neurons that are happier metabolically. All sorts of ways of showing this. Here's one version, a technique called silicon microphysiometry, measuring metabolism of cells in vitro in real time. Throw in an insult, metabolism crashes. Throw in an insult, overexpressing glucose transporters, and you buffer metabolism. So if you've gotten that far, what you better now be able to show is these are neurons better able at handling the glutamate and the calcium and the oxygen radicals, and absolutely that, overexpressed glucose transporter and less glutamate in the synapse, less calcium mobilized. And if you've gotten to that point, what you better now be able to show is fewer dead neurons. In vitro models in all these cases, in vivo taking advantage of reporter genes showing the more neurons infected, the smaller of a lesion, that's great. One additional thing you need to show though, which is most of us are not going to have the luxury to schedule our stroke for next Tuesday afternoon when the doctor's got an opening. If this stuff is going to work, it's predominantly going to have to be done after the fact. What sort of time window do you have? And this is explored here. Introduce overexpression of the glucose transporter at the time of an insult. You decrease the damage. This is a stroke model. First introduce it a half hour, post-insult protection 1 hour. By 4 hours, the window is closed. Just getting a sense of what sort of time period you got to work with. So over these years, we have now worked our way up and down various pathways of neuron death, 20, 25 different genes, asking, does it protect against this insult, that one? Do we understand the mechanisms? What's the time window afterward for expressing different of these genes? Okay, so all of that under our belt, we then began to be able to think about doing gene therapy against stress, gene therapy against the actions of glucocorticoids. Now, the first thing we wanted to do was be able to have a vector system that would only express during stress, a stress-inducible system. And what we began to think about then was a very clever, bizarre thing that herpes virus, which was our main vector system that we use, that herpes virus is able to do. We all know this. You get a herpes infection, you get a flare-up, it goes away, and then it goes latent for a month or 50 years, and then at some point it comes out of latency. When do you get a herpes flare-up? When you are under stress. Under stress like bereavement, like divorce, like chemotherapy, like social subordination in a rodent. All of those bring viruses like herpes out of latency. What's the logic there? Chronic stress, immune suppressed, great time to come out of latency, make a bunch of new copies of myself, and then go hide again. It's a very opportunistic thing that viruses like these have evolved. So thus you have the question, how is herpes virus able to know that the host organism is stressed? And a few years ago, a Harvard group got the answer to this. Okay, mechanism of action of glucocorticoids. Steroid hormone enters target cell, binds to steroid hormone receptor, at which point it translocates to the nucleus and binds, in this case, glucocorticoids to a glucocorticoid responsive element. Glucocorticoid responsive promoter, and it does its transcriptional thing at that point. What did these folks find? Herpes virus is able to measure your glucocorticoid levels. It's got GREs all over its genome. All of its genes of reactivation out of latency have GREs upstream of it. It knows when you're stressed. So we took one look at this and we wet our pants. We were so excited. We immediately made our vectors and stuck in a half dozen of these GREs in sequence there. And what we wound up getting was a stress glucocorticoid inducible vector system. In what you've got here, endogenous glucocorticoids, synthetic ones, not by the other steroids, dose response manner. Another hieroglyphic here in vivo, increasing circulating levels of glucocorticoids, more induction of this vector. Finally, a sustained seizure model, massive induction of this. You can use the system to decrease hippocampal damage after these insults, a stress-inducible system. Now, what we then began to focus on was what kind of genes we would want to express driven under circumstances of stress. And we made a family of 3 genes at this point. One overexpressing an enzyme that breaks down glucocorticoids. One expressing a transdominant mutant version of the glucocorticoid receptor. The one most pleasing to me is on the bottom. Okay, back to 30 minutes ago, that business about estrogen. Not only does estrogen not make things worse, estrogen is neuroprotective in all sorts of domains. Estrogen, being a steroid hormone, works the same way as glucocorticoids. Estrogen binds to estrogen receptor, translocates to the DNA to an estrogen responsive element, and then having beneficial transcriptional effects. So what we did was go and make ourselves an artificial gene that was half of a glucocorticoid receptor and half of an estrogen receptor. The hormone binding domain from the glucocorticoid receptor, the DNA binding domain from the estrogen receptor. What happens? Along comes a stressful period, binds the glucocorticoids, and then translocates to the estrogen responsive element instead of the glucocorticoid responsive one. And this winds up transducing an endangering glucocorticoid signal into a protective estrogenic one. One example of this, BDNF, the growth factor, all you need to know about BDNF is it's a good thing. You want to have a lot of it in your hippocampus. Stress drives down the levels of it, glucocorticoids drive it down, estrogen boosts up BDNF levels, and here we see estrogen treatment, up go BDNF levels. Now instead, here is a rat overexpressing this chimeric artificial receptor and stressed, and it suddenly acts as if it's exposed to estrogen. So here, being able to build not only a protective stress response that gets rid of glucocorticoid actions, but gets you something protective for free. Now, using this approach, we've now started to look at other issues. Not only can we use gene therapy strategies to protect neurons from neurological insults, can you begin to manipulate behavior? Can you begin to deal with animal models of psychiatric disorders? And here's one example of it, now overexpressing this anti-glucocorticoid system instead in the amygdala. If hippocampus is about learning and memory with mindless simplicity, the amygdala is about fear and anxiety. And what we've got here is, by blocking the effects of stress in the amygdala, you decrease fear conditioning. You can block fear conditioning, which is normally greatly facilitated by stress. You can do the same thing in the amygdala and decrease anxiety. Rats overexpressing these things are more likely to explore new open spaces, which they are normally quite averse to. With chronic stress, the amygdala expands. Remember back 20 minutes or so, stress causes the hippocampus to atrophy. Chronic stress, the amygdala expands, becomes more excitable, LTP is enhanced, you are more fearful, you learn fear associations more readily, and overexpress these anti-stress vectors, and you can decrease the size of the dendritic arbor. All of that, good. First baby steps of something in the way of gene therapy for psychiatric disorders. You could then take it one step further and forget blocking the pathogenic effects of stress. Can you now simply block some of the physiological but negative ones? Can you enhance cognition during stress? By now, a whole series of studies we have by manipulating the glucocorticoid system, blocking glucocorticoid receptor action, overexpressing another type of receptor that's beneficial, you can now generate a rat which, instead of learning worse during stress, learns better. Finally, that issue of neurogenesis in the hippocampus. As promised, with glucocorticoids with stress, you suppress neurogenesis, and what you see instead is you push towards an oligodendrocyte phenotype. When you are inhibiting neurogenesis in your neural progenitor cells in the hippocampus, you push instead in that direction, use one of these anti-glucocorticoid vectors, and you push away from the oligodendrocyte phenotype and push back towards neuronal production. In theory, you could now make a hippocampus which makes more neurons during stress instead of less. Okay, so all of this is wonderful. Hooray for us. Highly protective. Isn't this terrific? Next slide is meant to be extremely sobering. And this is a slide that I've been putting up proudly starting in the mid-90s or so, and somewhere a few years ago it struck me, in fact, this was quite embarrassing putting this up. This is an explant of human brain tissue infected with these herpesviral vectors of ours expressing a blue reporter gene, and you can see little bits of blue there expressing along the margins there. You can get these vectors to work in human brain tissue. And 14 years later now, this is still the best damn picture I've ever got to show. The field is not going anywhere yet in terms of being able to translate this into clinical medicine very soon. There's problems of delivery, there's problems of safety of the vectors, all the sorts of things that has hamstrung gene therapy in all sorts of other realms. Doing it in the nervous system is the final frontier in many ways. This is lurching in the right direction. So a little bit of good news there, but good news only, like at this stage, have a neurological insult. If you want to save 3 adjacent neurons, we can do it for you right now. You want to save 4 neurons, you're going to have to wait a while. But nonetheless, the field is moving in the right direction. Okay, so back to that slide before, putting up what can be done. Block the hormone release, block the receptors, block the downstream consequences. There's another thing you could have put up on that chart, which I failed to do because it is so idiotically obvious and simplistic that it's just absurd to put up, which is, ooh, don't get stressed. Why? That's terrific advice, and don't get stressed. What I will transition to now in the last part of this talk is looking at this issue of go finish this lecture, step outside and unexpectedly be gored by an elephant. You're going to have a stress response. You can't psychologically reframe your experience and grow from adversity or something like that. Go and have a concussive head trauma, you're going to have a stress response. Go and have any of those acute neurological disasters. Go and have the sort of ambiguous social interactions that we navigate every day, and only some of us will have stress responses. Only some of us will have different, less adequate degrees of coping with stress. And what I transition to here in this last section is beginning to deal with this issue of why do some bodies, and even more importantly, some psyches, deal better with stress? Individual differences. Okay, so you want to study this. You can study this in humans, where there's all sorts of fascinating things. For example, findings like major depression, and for many years afterward, you were more at risk for cardiovascular disease. Very solid finding by now. That's great, but God help you if you've got to understand all the intervening steps between those two events there. It takes forever. You outlive your thesis committee. It's very difficult stuff. Okay, so instead, look at these issues in rodents, lab rats, shorter lifespan, control the stressors, control the pathogens. A big problem there, though, is if a lab rat is a good model for your emotional life, you've got problems. So, picking something in the middle there, non-human primates, that's great, all the obvious similarities, but what's also clear is if you're dealing with captive primates, the potential for distorting behavior due to the captivity is enormous. So with all those things in mind, for the last 31 years, each summer I skip out of my lab and go and study these issues in a primate population, asking who gets the stress-related diseases, but studying primates in their natural habitat. And for these summers, these have been my study subjects. These are olive baboons, savanna baboons, living in the grasslands of East Africa. These are the baboons that live in these troops of 50, 100 animals or so. And this is their habitat, the Serengeti ecosystem. And this is your basic sort of Robert Redford, Meryl Streep, grassland mosaic, plateau, savanna. And what you have here is something critical for what I'm trying to study. If you are a baboon and you had a choice in the matter, this is where you want to live. Fabulous ecosystem. You live in these big troops, so the lions don't mess with you often. You have a lower infant mortality rate than among the neighboring humans. Probably most importantly, you only have to spend 3 hours a day getting your day's calories. Critical implication. If you only have to work at that 3 hours a day, you've got 9 hours of sunlight every day to devote to being just miserable to some other baboon. That's the critical thing. These guys are not getting stressed by predators or malnutrition. They are getting stressed by each other. Overwhelmingly, if you are a baboon in the Serengeti and you're unhappy, it's because another baboon has worked very hard to bring that state about. They are perfect models for westernized stress-related diseases because, like us, they have the luxury of generating psychosocial stress. So what is the stress about in their world? You go and study these guys, and within hours, what's clear is not all baboons are equal. There are instead dominance hierarchies, a number 1, 10, 20, and the quality of life varies enormously. It influences who grooms who. It influences what food you get access to. It influences your mating success to a certain degree. It influences, do you get to sit in the shade in the hottest part of the day? Do you get to sit in the highest branch of a tree? When the leopard's around, rank is very important. Rank is a very powerfully organizing feature of their society. To give you an example, here's a middle-ranking male who has just killed an impala. This is hot stuff. This guy is about to get 3 days worth of protein. This is wonderful. And suddenly, from out of nowhere, along comes the alpha male. And what we see is our guy picks up, and within seconds, the alpha male has the kill, exploiting the labors of the working class. And what you see here, most strikingly, is there was no fight. There was no overt aggression. The guy who left didn't even mumble dirty words under his breath. When the hierarchy is stable, these animals are smart enough to know their place in the society. Rank is a very, very stabilizing, defining feature of their world. So you look at this, and of course you wonder, well, what goes into being a high-ranking baboon? And for reasons I can explain later if there's time, 90% of my work over the years has been on males rather than females. What goes into be a high-ranking male baboon? One critical feature of it is to win the right fights. Male baboons have huge canines, bigger than an adult male lion's. They use them. They have the highest aggression rates of non-human primates. Here's a male with a deep canine slash in his shoulder, and this is not just ritualistic fighting. This is a male who had joined a troop of mine a few weeks before, and the only way of describing this guy is he had terrible political skills. He was hassling all sorts of guys he had no business going near. One night, coalition of 6 of them ganged up on him, and this is what was left in the morning. Over my 30 years, the leading cause of death of male baboons are male baboons. So extreme high rates of violence. Far more often, though, you get threats of violence. You get conventionalized gestures like this threat yawn, where it is clear what you're telling the guy in the other tree there is, look, you remember what happened last week? I've got the same teeth as then. Do us both a favor and don't push your luck. And most of the time, they don't push their luck. Instead, they give a conventionalized gesture of subordination. So violence or threats of violence. If that's all these guys were about, they would be boring. What they're mostly about, though, are things that look real familiar to us, things that have to be defined as psychologically stressing each other. So what is it that makes psychological stress stressful? Jumping over a huge literature, what you see is for the same external reality, we feel more stressed, we activate more of a stress response, we're more at risk for a stress-related disease if we feel like we have no outlets for the frustration caused by the stressor, if we feel like we have no control, no predictability, if we interpret things that things are getting worse, and if we lack somebody's shoulder to cry on. And baboons are brilliant at manipulating these variables in each other. First example. Here is a male baboon who is in a sexual consortship with a female. This female is in estrus, in heat, ovulating, and he's sitting there grooming her. He has been with her for the last 3 days. And you'll notice just happening to be 6 feet away on the other side of the bush is another high-ranking male baboon who just happens to have been 6 feet away around the clock for the last 3 days. Never an inch closer, never an inch further, not fighting with the guy, not even threatening him, just around. Just constantly present, just harassing the consortship. And this is enormously costly. This guy hasn't slept in days, he hasn't eaten. What you wind up seeing is almost half the time, this guy will pick up and voluntarily relinquish the consortship to this guy. This is not violence or threats of violence, this is grinding psychological stress. Next version. Here we have one of the wild card strategies for a male baboon, which is get yourself a coalitional partner to back you up in a fight. And if you've got one you can rely on, this is really great, and you can be unstoppable at that point. And here we see two of these coalitions facing off. And if you don't know baboon body language, these are four very uptight baboons. And what's clearly about to happen is violence about to break out. And a second later, yes, indeed, the fighting begins. Anybody see the fourth baboon? There he is. There he is, showing this close relative of ours, which is when the going gets tough, you bail out of there. About 40% of the time, when the fight occurs, your erstwhile partner bails out on you. And of the times that he does, a third of the time, he joins the other team. Talk about psychological stress. You go into a fight that's life-threatening, and you don't even know who's on which side. Final version. Here we have a male, again in a sexual consortship with this female, grooming her, and in this case he's being harassed by this guy, high intensity, a foot away, threat yawning him in his face. This guy in the middle is under stupendous pressure. You know he is going to crack, he is going to become violent, and a second later, yes indeed he does, he spins around and he bites the female. Oh. Another feature of this close relative of ours, which is when the going gets tough, look at somebody smaller to take it out on. About half of baboon aggression is third-party aggression dumping on an innocent bystander. A male loses a fight, chases a subadult who bites a female, who slaps a juvenile, who lunges at an infant. What does this mean? You're sitting there minding your own business, bird watching, and some other guy's having a bad day, and it's your rear end that gets slashed. Lack of control, lack of predictability. So looking at all of these traits, what goes into being a high-ranking individual in this sort of world? Yes, being big and muscular and big canines and all that sort of thing, but also all sorts of social intelligence stuff. Which coalitions you form, which ones you don't go near. Impulse control, which provocations you could ignore, as opposed to getting into every fight that eventually leaves you crippled. And starting with these guys in the 70s, it struck me watching them. Another feature of this was successful males in a world like this will have bodies that deal well with stress. And that's what I proceeded to go and study, and what I'll show you is I then wasted my next 15 years studying that, because it turns out that was a stupidly simplistic question to ask, because there's something much more interesting happening. Okay, so how do you study this? What does your social rank have to do with your stress physiology? Part of what you do is your basic Jane Goodall scene of collecting behavioral data, but what you also then have to do is get physiological data out of these guys with all sorts of constraints. And this is a frame from a PBS thing a few years ago, and you could see in this frame a white flash there on this guy's shoulder, a blowgun dart exploding at that point. All sorts of constraints. You got to get an anesthetic that doesn't mess up the hormone you're trying to study. Next, you got to dart everybody roughly the same time of day and season to control for rhythmicity. Next, if you're trying to get a first blood sample out of a guy that tells you something about nice resting levels of stress hormones, you can't dart somebody if he's had a fight that morning, if he's injured, if he's sick, if he's mated. You want to look at cholesterol, you can't get somebody if he's had breakfast yet that morning. Next constraint, you can't dart somebody if he knows it's coming. There can't be any anticipatory stress. So you can't just get in your Jeep and chase the guy around for 3 hours, and when he's winded, you suddenly nail him. Final constraint, if you want to get that first basal sample, you've got like 1 to 2 minutes from the time the anesthetic kicks in to get a first sample out of the guy. So given all those constraints, about once a decade it works flawlessly, and you've got an anesthetized baboon at that point. You take blood, CSF, tissue biopsies, you can have a fancy heparin-locked syringe on your acacia branch. You get all your samples there. Samples are processed on a hand-crank centrifuge, which is as awful as it sounds. Guy recovers overnight in the cage, let him go the next morning to his buddies, get your dry ice shipment once every 2 weeks, keep your samples frozen, sneak them through customs, and you're all set. Okay. So given this, what have I looked at over the years? What's going to be the first thing I look at? What's the relationship between rank and glucocorticoid levels? And by now, you can absolutely define the perfect profile. What do you want? Absence of stress, you want like 0 glucocorticoids in the bloodstream. Along comes a psychological stressor, don't get suckered in by it, keep the same low levels. Along comes a real physical stressor, have the biggest, fastest response possible, and the second it's over with, have the biggest, fastest recovery you can get. And that's exactly the profile that you don't see if you're a low-ranking baboon. Everything in them is worse. Basal levels of glucocorticoids are elevated. They're elevated even in the absence of stress, because if you're low ranking, you're always stressed. You spend 2 minutes digging some food out of the ground, anybody can rip you off. You spend the whole morning getting somebody to groom you, anybody else can disrupt it. You're sitting there, you get your rear end slashed by somebody else. Lack of control, lack of predictability, lack of outlets. When a real stressor finally does come along, subordinate guys have a sluggish stress response. When it's over with, they have a sluggish recovery. Exactly what you don't want to have. And I then spent the next dozen summers out there sort of doing circuitry on these guys, trying to understand what's working differently according to rank at the level of brain, pituitary, adrenals, that accounts for these elevated glucocorticoid levels in subordinate baboons. And mechanistically, what you turn out to see is you get hypercortisolism in a subordinate baboon for the same exact reasons that you see elevated glucocorticoid levels in a human with major depression. Cognitive psychology soundbite: depression is learned helplessness. In lots of ways, being a subordinate baboon is an adventure in learned helplessness. Okay, so using this approach, working up and down all sorts of different physiological systems, and you always see the same thing. It works completely differently depending on your rank, and consistently, it's the low-ranking baboons who are more set up for stress-related disease. So this seems really straightforward, which is, go out and be a big high-ranking baboon and fight and win, and one big social Darwinism blowout there. Let me spend the last couple of minutes, though, showing how this is totally wrong and how there's much more interesting stuff going on that is much closer to home. Yes, your social rank is important, but first qualifier, far more important, is the sort of society in which this occurs. One example among many. All the data I just summarized for you come from hierarchies that are stable. What do I mean by stable? Number 5 wins 95% of his interactions with number 6. Number 5 loses 95% of his interactions with number 4. They're stable dyads. On the other hand, if number 5 is winning only 51% with number 6, they might be just about ready to flip there to the other way. That's an unstable dyad. All the data I showed you come from seasons where more than 90% of the interactions reinforce the status quo. Every now and then, something happens. Some critical male dies, is injured, leaves the troop, joins the troop, who knows what, and something destabilizes, and the hierarchy completely comes apart for 3 months. High-ranking guys are still higher ranking than the lowest ranking, but among the highest ranking cohort, it's chaos. Everybody's fighting, nobody's mating, nobody's eating, the trains don't run on time, enormous amounts of conflict there. Eventually it settles down. What we see here is high-ranking males during a stable period, nice low basal glucocorticoids, same males of same average rank during an unstable period, and the advantages go down the tubes. It's not being high-ranking, it's being high-ranking in a stable hierarchy because you've got all the psychological advantages there. Be high-ranking, be in the Tsar's palace while the revolution is happening at the gates. That's not a very good place to be. When the hierarchy is unstable, it's the highest-ranking individuals who now show the worst health. So it's not just your rank, it's what the rank means. Next, it's also how you personally experience this. 2 examples, actually, interest of time, 1 example. Okay, this was some years ago. An extremely aggressive male joined the troop who, no doubt trying to impress the other males, spent all of his time attacking various females. And 2 weeks after he joined the troop, I was able to keep track of how many times had females been attacked by him to the point of his drawing blood, ranging from females who were lucky and those who were attacked most frequently. Then I darted a whole bunch of these females and looked at their white blood cell counts. Just Neanderthal immunology, but the best that could be done out there. And what you see here is a dose-response relationship. The more often they were attacked by this guy, the more immunosuppressed they were. Here were white blood cell counts in the females before the guy joined the troop. So you say, ooh, what are the effects of a destabilizing aggressive male and immune function female baboons? The answer is, it depends. It depends on your personal experience of it. If you're lucky enough to be sitting on the sidelines, it has no effect whatsoever. It's not the abstract state of being in a violent society. It's how often is your own personal nose being rubbed in it. Final variable. And this is the strongest one. Yes, rank is important, as well as its meaning, as well as personal experience, but most important probably is your personality with which you filter all of this. Now, 20 years ago, if a primatologist said the word personality, they would instantly have their tenure taken away. This is now the hottest term in primatology. Non-human primates have stable, long-term differences in temperament. And a number of years ago, what I did was sit down with 15 years of behavioral data and try to come up with every personality marker I could think of in these baboons. You anthropomorphize like crazy. What's a baboon's equivalent of a hobby? What's a baboon's, smiling on the right side of the street? All that sort of thing. Came up with like 50 measures, controlled for rank, controlled for stability of rank, ran it against the physiology, and out came an interesting finding, which is all of the good physiology you get if you are a high-ranking male in a stable hierarchy has nothing to do with being high-ranking. It is all due to some personality styles that tend to go along with high rank. And you could be number one in the troop, and if you don't have any of these styles, you're going to be just as hypercortisolemic as number 20. So the final question here becomes, what personality style works for a baboon? And given that I'm from San Francisco, I could spend my weekends in like Marin giving hot tub seminars to baboons, teaching them how to manage stress by being exactly this way. How does a male baboon go about competing with other males? First variable, can you tell the difference between threatening and neutral interactions in the world? What does this look like? You got a male, he's sitting there, his worst rival on the whole planet shows up and threatens him in his face from a foot away. This is bad news. What do you do? What do you do? You stop what you're doing, you take this defensive, vigilant stance. Sitting there, worst rival on the whole planet shows up and takes a nap 100 yards away. What do? You do whatever you were doing. This is not a big deal. Pathetically, your average male baboon does not behaviorally distinguish between being threatened in the face by his rival and his rival napping. It makes him just as disruptive in his behavior. This is type A personality. This is sitting there saying like, look at that guy, I hate him, and look at the way he's snoring, and he does that just to get on my nerves. This is seeing stressors that other individuals don't. After controlling for rank, if your behavior is just as disrupted by your rival sleeping as threatening you, twice the glucocorticoid levels in your bloodstream. Next variable, the guy is threatening you in your face. What do you do next? Do you abdicate control and let him start the inevitable fight, or do you at least get some control in this circumstance and be the one to start it? Two different ways of measuring this, but showing males who abdicate control, much higher levels of glucocorticoids. Next variable, you've had the fight with the guy. Can you tell whether you won or lost? Oh, this is rather fundamental. And pathetically, again, your average male baboon has the same likelihood of what behavior he does afterward, depending on, regardless of whether he's won or lost. If you can't tell the difference between a promotion and losing, you're going to have significantly higher glucocorticoid levels. Finally, you've had the fight and you've lost. What do you do next? Do you go and mope by yourself, or do you go and beat up on somebody smaller? Okay, how best to frame this? Male baboons who lack social outlets and go and mope by themselves, significantly higher glucocorticoid levels. Look at this. Can you tell the difference between big things and little things? If it's a big thing, do you at least get some control? Can you tell if the outcome's good or bad? And if it's bad, do you have some effective coping outlets? People pay to be taught to do this sort of thing. And you go back in the records of these guys, these low cortisol guys, and you look at them back when they were juveniles, and they were like this back when. It's a long-term stable trait. Final marker, independent of rank, stability of rank, male-male competitive style, etc., the single best predictor of having elevated stress hormone levels is being socially isolated. How often do you groom? How often do you sit in contact with someone else? How often do you hold an infant? All of this being highly predictive. Okay, so what do we got here? First third of the talk, extremely depressing. We are now 60, 70 years having to think about stress as playing adverse roles and how all sorts of outposts in our body function. Almost certainly, we need to think about stress playing an adverse role in the consequences of some of the most common of neurological disasters and how we age and how some of us will age cognitively more successfully than others, and increasing reasons to think this is quite pertinent to our own brains. Amid all that bad news, one version of the good news has shown you at the more reductive end the capacity to understand the nuts and bolts of what goes wrong to begin to design rational interventions, gene therapy, and as we all know, that's just the tip of the iceberg of some things that could hopefully be done down the line. There is hope in that realm. Final third here, at this more integrative level, when looking at the sort of stressors that do us in, these are not the ones where everyone will catastrophically have a stress response. This is the world of us having these individual differences that are supremely sensitive to our psychological makeups and our social contexts. And basically, that's the place to stop here. Because insofar as we are not getting ulcers because we are chased by predators or having to wrestle people for canned food items in bombed out supermarkets, insofar as we live well enough and long enough to pay for these things, insofar as we are smart enough to invent this stuff and foolish enough to fall for it, all of us have the potential, I think, to be wise enough to keep it in perspective. So on that note, let me acknowledge the members of my lab who've done this work and my field collaborators over the years out in Kenya, including my wife, and thank you for your attention.
Given the hour when we suggest that people ask you questions in the reception, is that right?
Yeah.
Thanks, Robert. That was just superb. Given the hour, I think perhaps we'll adjourn to the south lobby for the reception. So if you have questions for Robert, he will be there to address them. So please do come to the reception. Thank you all very much.
Well, it helps to be a manic.